Hello, Biology 1 students. Mitch Albers here. In this Biocast, we're going to take a look at some of the important aspects of the biological molecules of life. So let's dive in. Let's say on the upcoming exam, you were given these structural molecules. And maybe the question was, can you identify them? Or maybe to zoom in on one specific and say, could you identify this part of the molecule that could be a functional group? Could you identify some of the behavioral characteristics that the molecule might possess based on its structure? These are all the kinds of things that you've been reading and learning about with the different biological molecules in this class. And so, we're relying on using some of our basic chemistry background, and we've introduced you to a little bit of inorganic and organic chemistry, and now we're getting into more of our discussions that will lead us down the trail of biochemistry. So what if you were asked the question, can you organize these molecules into groups? Okay, so the first task would be, let's say, can we identify which ones are the sugars, the carbohydrates? Okay, so let's see if we can do that. So we'll just start up at the top, start with the first structure here. Okay, this molecule here, I identify that as a sugar. We've been talking about sugar molecules having long carbon chain backbones, lots of hydroxyl groups, carbohydrates, hydrates. That looks to me like a sugar. Okay, there's another molecule just down close by, looks very similar, almost identical. Almost, that's the key. Not identical, but almost. Another sugar. Okay, continue on. I know that sugars can form rings. Okay, and sometimes they can appear as chains of carbons, and also in a ring form. So that looks to me like it might be a sugar. Continue on. Well, I hear there's another sugar that's in a ring form. Okay. And now I'm seeing some more that kind of using that plan of rings. I'm seeing rings linked together. Looks to me like a type of sugar. And also this one down below here looks to me like a sugar. Okay, so we could group all of these as carbohydrates. Simply, they're in the group of sugars. Okay, so now let's look for the molecules that might appear as fats, would go into making up fats or the various lipids. Okay, I would identify this one. I identify this one. And then this great big one here. And you're going to be asking maybe, well, how is he able to define that these are sugars and lipids and so forth? Well, when you study organic chemistry and learn the basics of life, you have to understand some of these molecules and the roles that they play. And when you really study their structures, they're going to tell you a lot about how they function. Okay, so these three that I've identified in red are the lipids. Okay, commonly known as fats. And all right, so then the next group here, let's try to find what goes into making up a protein. So let's see if we can identify in green. Okay, I believe this molecule goes into making up proteins. I believe this molecule can, and I also believe this molecule can. And you're kind of wondering, well, how would you know that? Well, there's one thing that I see that all three of these molecules share in common. They have a grouping like this and a grouping like this. And all three of them have the same plan. And I know that the building blocks, the monomer groups, of the amino acids have that basic plan. So we're going to put those into the proteins. Okay? And then the last group here are the nucleic acids. And the nucleic acids are going to be our DNA and RNA molecules. And I don't see any DNA, I don't see any RNA, but when I kind of think about, well, so what goes into making up DNA and RNA? Well, I remember that the basic monomer group is called a nucleotide, and when I think about it, what goes into making up a nucleotide? Well, if my memory serves me right, a nucleotide consists of a phosphate group, a 5-carbon sugar. So here we've got a sugar that it's a sugar, but it also goes into making up nucleic acids, so we're going to share that group. And then what fills up the rest of the remaining part of a nucleotide is a nitrogenous base, a carbon-nitrogen ring structure. There's various forms of this base, but this is one example. And those three together make up a nucleotide, which go into making up nucleic acids. Okay? And so the only structure I'm left with here that I haven't been able to group into one of those 4 macromolecule groups is the last one here. And this one, okay, so I'll just kind of put a scribbly line around it, wiggly line. This is the structural formula for, what is it, H8 butane. And it's a gas. And what could we say about that molecule? Well, one of the things we talked about in the carbon chapter was that when we have a structural arrangement around a double bond, like we're seeing between the 2 carbons here, okay, and if these groups up here are on the same end, this is called the cis position. But if one of these, let's say, were to transfer down here, then it would be in the trans position, okay? And for the molecular formula C4H8, there are various forms, structural forms of that molecular formula, which you know better as isomers. Okay, so I wouldn't probably expect that you, unless you took organic chemistry, that you knew that was butane. But I would, when you look at the structure, and just as I'd mentioned, you know, it does fall into a class of isomers, which we talked about, which are the cis-trans isomers. So let's take a look at these different groups and see what are some of the characteristic structures and functions that we're going to expect from them. All right, we'll start with the carbohydrates. So from that main group, I've just clustered then the molecules that were on that previous slide here now under carbohydrates. So what are the kinds of things we could start looking at these molecules and making some general statements to understand both structural aspects and functional aspects. Well, one of the things what we want to do is count the carbons, okay? And so for this molecule here, if we count the number of carbons in the chain, I see 6 carbons. So it's a 6-carbon sugar. Likewise, I see here a 6-carbon sugar. Okay, and then when I go over here, wherever I see an intersection of 4 lines, I know there's a carbon in every one of those locations. There's a carbon here, and I count those carbons. I see 6 carbons in that sugar. Okay, all the 6-carbon sugars are referred to as hexose sugars. You can always identify a sugar because it ends in the suffix OSE. Hex is meaning 6, so these are 6-carbon sugars. Okay. Now this sugar way over on the right here There's a carbon there, 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 and there, and there. Or excuse me, this is an oxygen, not a carbon. So carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. So this is a 5-carbon sugar. Okay. So that's in a group of sugars that are called the pentose sugars. And if my memory serves me right, that coming off carbon 2, if I've got a hydroxyl group coming off there, so I'm identifying this grouping here, that's a functional group, which is called a hydroxyl functional group. Okay, that this might identify that sugar as ribose. Okay. I know that because there's another 5-carbon pentose sugar called deoxyribose where it's missing that sugar or that oxygen off carbon 2. Deoxyribose means less sugar. We might run across that up here in our discussion here when we get to nucleic acids. Okay, some other things that I might be takeaways from this. So again, carbohydrates are carbon molecules with hydroxyl groups. Lots of hydroxyl groups. Another functional group we're seeing on the sugar here is a carbon double bond oxygen. That's called a carbonyl. Carbonyl group. And if that carbonyl group is located on a N carbon, like it is here, and we've got the same thing happening here, these two sugars fall into a group which are called aldose sugars. Or sometimes we call them aldehydes. Okay. So those are the kinds of things that you can kind of just by looking at the structure, knowing something about them and getting some information about them. Okay. Here I've got 2 sugars linked together to make up a single sugar. That's going to form what's called a disaccharide. Type of sugar. And then we've got a sugar here that's made up of more than 3 sugars in a chain, and we would call that a polysaccharide sugar. Okay. Now, we'll get into some of the details of disaccharides and polysaccharides, but let's start simple and start with the monosaccharide sugars. First of all, you know, carbohydrates, when we think of functions of carbohydrates, the 2 main functions are going to be energy source and structural. Energy source and structural are going to be our primary functions for the carbohydrates, the sugars. All right, so let's take a look at monosaccharides. So from the, again, from the list that we started with, now I'm narrowing it down from all the carbohydrates, the molecules that were on that original list that are now monosaccharides. Okay, so let's look at some characteristics here. Okay, earlier we counted this sugar as a 5-carbon sugar. Pentose sugar. Okay, we saw these 2 sugars here as 6-carbon sugars. Actually, all 3 of these, the ring one as well. And we classified those as hexose sugars. Okay, so these are all monosaccharide sugars. They're single sugars. And the monosaccharides are going to become the monomers or the subunits for building the larger carbohydrate molecules, the disaccharides and the polysaccharide molecules. So, so let's take a look at how, first of all, we see some variations in these structures. We see chains in these sugars, and we see rings. Okay, and so unless you've really studied the structures, you could identify these two chains. One is glucose, and the other is galactose. Okay, glucose sometimes can be called also dextrose, and it's often referred to as blood sugar. Galactose, a monosaccharide sugar that goes into making up lactose, which is a disaccharide and is commonly found in milk sugar. Okay, but if you study glucose and galactose, they have the same molecular formula. They're both C6H12O6. Okay, and so they are both isomers. They have the same molecular formula, but they differ structurally. And if you study them, they almost look identical. The only difference is the orientation of this hydroxyl group here of carbon 3 and here on carbon 3 between the two sugars. That's it. But that little difference makes all the difference in the world as far as their behavior and what their properties are. Okay? Always structure determines function, and we harp on that quite a bit here in biology, and particularly with these biological molecules. Okay? And then down below, the ring structure here, this also is glucose, but it also is the ring form. So let's talk a little bit more about these chains and rings and figure out what's going on there. So, first of all, when we're talking about the biochemistry of life, we're going to probably be focused mostly on how these biological molecules are, what they're doing in our cells. The cell is the basic unit of life. And so these molecules will be present in our cells. And you have to think about, what's inside a cell. What mostly makes up the cell environment? Well, the cell environment is going to be described as an aqueous environment. And what that means is that it's a water environment. It's consisting mostly of water. That's what makes up most of the cytosol within our cells. And dissolved in that watery environment, that aqueous environment, are going to be various sugars, fats, and proteins, and so forth, all these biological molecules. And these sugars that can exist both as chains and as rings. Okay, so we have a chain, carbon chain form, and a ring form of the sugar. But when this chain goes into an aqueous environment, the tendency is that it's going to form a ring. So we have to kind of understand that this is the tendency that these structures are going to be, is taking on this ring-like structure. Okay? Because we're going to be talking about these molecules in living cells and organs and systems, and so they're most likely going to be occurring in cells that are in these aqueous environments. So just like we do over here, we can count our carbons. We have carbon 1, carbon 2, carbon 3, carbon 4. And remember, on the ring, wherever there's an intersection of 4 lines, the only atom that has a bonding capacity of 4 is carbon. Carbon can share 4 pairs of electrons. Each covalent bond represents a pair of electrons that are being shared with the other atoms around it. Okay? So we got carbon 1, 2, 3, 4, carbon 5 right here, and then the 6th carbon up here, carbon 6. Okay? So we're going to have to start getting used to just seeing a lot of these more these ring structures of these sugars from here on out. All right, so now that we kind of understand the basics of, just understanding that both the chain and the ring form of these sugars exist, and they tend to form the ring structures in cells, let's start linking sugars together and building new sugars. Okay? So, let's build a disaccharide. Okay? So again, a di meaning two sugars. Okay, a monosaccharide plus a monosaccharide yields a disaccharide. Okay, so here we're looking at glucose plus a glucose, which then yields maltose, a disaccharide sugar. Malt sugar is the, you guys know this. This is the sugar you commonly find in beer. Okay, so if you drink a beer, you're ingesting maltose, which is a disaccharide, and you're providing some carbohydrate then in your diet if you're drinking a beer. Okay. So now, how did we link the two sugars together? Okay, well, that process, as you recall, is dehydration synthesis. Coming off carbon 1 here is a hydroxyl group. Okay, coming off carbon 4 of the adjacent sugar is also a hydroxyl group. So this OH grouping is a hydroxyl group reading it this way. This is a hydroxyl group reading this way. Okay, so what we're going to do is we're going to take one of those hydrogens and one full hydroxyl group and remove two hydrogens and oxygen to form water. Okay, so in doing that, we are dehydrating this molecule. So this is dehydration. And now we've created something new. We've created this maltose sugar. Okay, so we've now created something bigger. We've synthesized a new molecule. So we're going to call this dehydration synthesis. Okay, and dehydration synthesis is going to be the basis of how we're going to build all the polymers, whether it's the carbohydrates, the lipids, the proteins, and the nucleic acids. So we'll see that theme play out. Okay, now just to mention, there's two other disaccharides that are out there. If you take glucose and link it with galactose, and we saw galactose and glucose in the previous slide, you can produce the disaccharide lactose. Okay, you know that as milk sugar. You can also take glucose and link it to the third hexose monosaccharide sugar that exists out there, and it's called fructose. And that yields sucrose, a disaccharide sugar. And you know sucrose is your table sugar. C&H sugar, that white sugar you buy, that's table sugar. That's what you sprinkle on your Wheaties in the morning. That's a disaccharide sugar. Okay, so there's 3 disaccharide sugars. You should be able to know what goes into making them and how they are put together through dehydration synthesis. Okay, so now let's build even a bigger carbohydrate. Let's take a look at how do we construct a polysaccharide. Okay, so poly is a term that means many. Okay, and saccharide, you know, is sugar. Okay, so when we link many sugars together, we form a polysaccharide. Now, So the classification that we've talked about here, we have monosaccharide. Okay, those are the single sugars made up of a single individual sugar. We have the disaccharides, okay, are two sugars. And then there's a group between the disaccharides and the polysaccharides, and it's called the oligosaccharides. And that usually are sugar molecules that range maybe from 3 to, well, it varies. It may be 10 to 20. So let's just say 3 to 20 monomers, individual sugars linked together to make up oligosaccharides. And polysaccharides are going to be the really big types of carbohydrates. And the polysaccharides in the sugars are sometimes referred to as the complex carbohydrates. Okay? And these are going to include things like starch, glycogen, cellulose, chitin, just to name a few. And these are all ones that your book refers to. Okay? So in the polysaccharides, individual glucose molecules are the monomers. Of building not only the disaccharides, oligosaccharides, and now here in the polysaccharides. Okay, so this large molecule here is our polysaccharide. Okay, this molecule down here is an example of a polysaccharide. It's a part of a polysaccharide. It's not complete. Either is the one in the middle. Okay, but what we're going to do is to link. Let's say here I've got 1, 2, 3, 4, 5, 6, 7, 8. When I want to link 8 glucose molecules together to form a polysaccharide chain, well, I'm going to do that through my dehydration synthesis. My target here is going to be those functional groups. I'm going to take a hydrogen and a hydroxyl group, and I'm going to remove a water here. And I'm going to link those two sugars together. Okay? And then I'm going to take a hydrogen here and a hydroxyl group here, and I'm going to remove a water here. And so between every one of these sugars, I'm going to remove a water. So in the synthesis of this polysaccharide molecule as an example, okay, I'm going to also produce, so I had 8 monomers, so I'm going to end up producing 7 waters in the process as well, so that they all then link together with a sugar bond. Okay, and when you're linking sugars together, you're forming what's called a glycosidic linkage, or a glycosidic bond. It's just a covalent bond, and glyco means sugar. Okay, so that's the kind of bonding we're seeing happening here. Now we can also specify. Okay, so let me back up for a moment. Let me just clear this out. Let's just go back to the previous slide for just a moment. The disaccharides. Okay, so when we put the disaccharide together, right? We said this was carbon 1, carbon 2, carbon 3. Here's carbon 4. Okay, on the other sugar, this is carbon 1, this is carbon 2, carbon 3, carbon 4, carbon 5, carbon 6, so forth. Okay, but if I'm going to take this hydrogen and this hydroxyl group through dehydration synthesis, okay, then I'm left with a bond between the oxygen coming off this carbon and this carbon here. And I'm going to end up linking carbon 4 to carbon 1 here. So we will specify this as a 1,4 glycosidic linkage. Okay, and that's what we're seeing here. Okay, so remember, this is carbon 1, this is carbon 4. So now our bond here is a 1,4 glycosidic bond, or 1,4 glycosidic linkage, however you want to specify the name. Okay, so that's what we're seeing when we're building a polysaccharide. Down below here, we also have a polysaccharide. Okay, now one of the things you should know about glucose, okay, is that coming off carbon 1, we have a hydroxyl group. Okay, and then a hydrogen coming off here. Okay, there actually is 2 forms of glucose. One is called alpha glucose, and the other is a beta glucose. Okay, and the only difference between the two is the orientation of that functional group. Okay, in the beta glucose, if I drew just the ring structure here, okay, there's a carbon here. So on alpha glucose, the hydroxyl group is in the lower position. It's in the plane of the carbon or the ring. It's oriented in the lower position. Okay, off carbon 1. In the beta glucose, that hydroxyl group would be up here, above the plane of the carbon in the upper position. Okay, so those 2 variations can occur. And I'm telling you that because when we look at a molecule, let's say these 1,4 glycosidic linkages that we just kind of explained, these are commonly found in starch. And starch is a polysaccharide that we take in our diet, and it's a really good energy source for us. And that's because we have the digestive enzyme, okay, that can get in there and weaken and break that bond so that we can hydrolyze it to split them apart. That's the reverse of dehydration synthesis. Okay, now I'm mentioning that because over here we're looking at this is just a short sequence of the sugars, the monomers that go into making up cellulose. Okay, and what we're seeing here, and I want you to take notice up here in this glucose molecule, you'll see this oxygen. Okay, so that oxygen is up at the top of the ring. Okay, now you'll notice some of the sugars in this structure, the oxygen is up at the top. It's up at the top. Okay, now you'll notice these 2 sugars, this one and this one, it looks like is the oxygen at the bottom. Well, no, it's still at the top. It's just that those sugars are oriented upside down in relation to the others. You'll notice in starch, all of these sugars, the oxygen is in the top of the ring. Okay, and I'm talking about these oxygens. Okay, but in this structure, every alternating sugar molecule, glucose, has the oxygen up or it's down. Okay, and that determines a lot of how these sugars are going to link together. And we end up getting the similar type of linkage, a 1,4 glycosidic linkage between the 2 glucose molecules, but now we're using beta glucose. And unfortunately, we don't have the digestive enzyme that can identify this particular bond. Okay, and so if we had that enzyme, we could get in there and hydrolyze and break that bond, but we don't. We lack that enzyme, and therefore we don't digest cellulose. Cellulose goes right through us, and cellulose, these long polysaccharide chains of beta glucose molecules that are linked together, will hydrogen bond with other cellulose polysaccharide molecules, and they will form these fibrous structures that go into making up the cell walls of plant cells. Okay, and so we ingest plant material. We're ingesting plant cells that are surrounded by plant cell walls, and when we try to break down the cellulose, we can't. We can't get in and break it up or digest it. But the cellulose does go through our digestive system, and we commonly call this the fiber in our diet, due to the structural nature of how these polysaccharide cellulose molecules form. Okay, so a lot of information just by looking at these molecules that are takeaways here. Okay, so let's continue on here. Let's go on and talk about the lipids. Okay, so we know lipids are commonly referred to as fats. Lipids play a number of functional roles. They're good for energy. They're good for insulation. They're involved with protection. They're chemical communication, because some of the lipids act as hormones, so they're involved with communication, cell-to-cell communication. So they're fairly diverse, and then they're also very important structural components that make up our cell membranes. Okay? So from that original group of molecules that we started off with, I identified this molecule goes into the lipids, this molecule, and this molecule. Okay? So and if you studied lipids, you should understand, identify, for example, this structure over here is what's called glycerol. Okay? This structure in the middle is a hydrocarbon chain. Hydrocarbon. It's a chain of carbons and hydrogens. And on this end has a carboxyl group. Okay? And we commonly will call these fatty acid molecules. Okay? And there's a number of ways you can see these molecules written. You might see something written like this, where the N carbon has a carboxyl group. Okay? And then we might simply just write this as CH2. 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 Okay? So right here, this is a CH2. CH2. Okay? CH2. That's the third one, carbon, that I highlighted there. Okay? And we can keep going on until we get to the N carbon right here, and on the end we will end with a CH3. Okay? So that's that. So that's one way we could indicate that. We also could indicate this with as a carboxyl group on the end. Okay? And then we simply could indicate this like this. Okay? And so all this line of points or bends, okay, wherever there's a bend, there's a carbon. Okay? And wherever there's an end, right here, there's a carbon. Okay? And then if we just use our bonding capacity, so that's just an abbreviation that tells us that that's a hydrocarbon chain. Okay? So we can put a hydrogen around these two carbons. So that's a CH2, just like we had above. Okay? CH2. I don't have to write the hydrogens in, but that would be the next CH2 grouping. and so forth. We just keep going down the chain. Okay, then we get to the very end, I'll end up with my N carbon having 3 hydrogens, which is my CH3 group, which is what we saw right there. Okay, so that's my typical fatty acid. The other thing we could take away from this, in this particular example, is that we're dealing with a fatty acid that is saturated. The fatty acid, the carbons are saturated in hydrogens, every carbon. Okay? The only way that we could make it unsaturated is if we put a double bond between those carbons. That would eliminate a couple of the hydrogens being saturated with fatty acids, and then we could classify it as unsaturated. And then when we add that double bond, that changes the angle of the hydrocarbon chain. And when it bends, that's going to allow the fatty acids not to fit as tightly packed together, and structurally that's going to make the fatty acid not solid at room temperature like the saturated fats are, things like in butter and animal fat, and so unsaturated fatty acids will in our diet are much better because they don't solidify, they don't tend to cause the problems in our cardiovascular system like the saturated fats do, leading to things like high blood pressure, strokes, and heart attacks. Okay, and then the last molecule to identify here is it's similar to what we just has similar components. Okay, it has glycerol here. Okay, just like we saw over here. That's the glycerol portion of this molecule. It has 2 fatty acid hydrocarbon chains. Okay, and then it has a negative charge phosphate group and a positive charge what's called choline group. And this makes up what's called a phospholipid. Okay, and we're going to talk about phospholipids, and then when we add glycerol with 3 fatty acids, we're going to construct what are called triglycerides. So let's take a look at triglycerides. So a triglyceride is just telling us that the molecule consists of 3 fatty acids linked to a glycerol molecule. Okay, so this is our glycerol, and these are our fatty acids. And so we got 1, 2, 3. And then we're going to construct our triglyceride. Okay. Now, the way that we're going to build our triglyceride is the exact same way we link the sugars together. Okay. We're going to grab a hydrogen off this hydroxyl group from glycerol, this hydroxyl group from the fatty acid. We're going to remove 2 hydrogens and an oxygen, which will form water. And we will then covalently bond the fatty acid to the glycerol. And then we're going to form a specific type of linkage, which is called an ester linkage, or an ester bond. Okay, and since we have 3 fatty acids, we're going to do that same process. We're going to actually form 3 molecules of water when we link all 3 of them together. Okay, and so we'll also produce 3 ester linkages, or 3 ester bonds, linking the 3 fatty acids to glycerol. And as I mentioned, this triglyceride molecule, this is the type of fat that goes into animal fat. So if you gain weight, you're adding triglycerides to your adipose tissue. When you eat bacon or and certain cuts of meat like steaks, if there's a little bit of animal fat on the edge, and you cook it and you eat it, you're taking in that type of fat in your diet. Okay, and I mentioned earlier, these fatty acids are all saturated fatty acids, and since they're linear chains, they fit very tightly together, and saturated fatty fats will pack tighter together and therefore will be solid at room temperature. Okay, so triglyceride molecules, also you'll notice that you don't see any polarity whatsoever. Okay, there's no charge anywhere on these molecules, so they have no polarity. And so we're going to say they are neutral. Since they have no charge, they're neutral. And behaviorally, when we relate them to water, they're going to be hydrophobic. Okay? Because of all those hydroxyl groups in sugar, and oxygen and hydrogen not sharing their electrons equally, the sugars have lots of polarity and are hydrophilic and dissolve in water very easily, but the triglyceride molecules do not. Water and oil, when we say water and oil, we're saying oils are in this group, okay, because some of the unsaturated fatty acid triglyceride molecules are oils, and they don't mix because of this neutral, no charge situation in the structure of the fat. Okay, so triglyceride molecules. Let's move on and talk about phospholipids. Okay, so like the triglyceride molecule, we have the molecule here that has the glycerol, the 3-carbon glycerol end of the molecule. Okay. And then, again, 2 fatty acids. Okay, so these are hydrocarbon chains, just CH2, CH2, long chains of hydrocarbons. And again, we simply call them fatty acids. Okay, then they have this negatively charged phosphate group. Okay, and then they have above this group here, which is referred to as choline, and it ionizes and is a positive charge. Okay, so we often refer to the choline group, I'm just going to change color here, and the phosphate group, kind of the head region. So we'll commonly see phospholipids kind of drawn like this, where the phosphate group and the choline are what make up the head of the phospholipid, and then the fatty acid hydrocarbon chains make up what we refer to as the tails. Okay, these tails, because they're made up of fatty acids that are neutral and no polarity, are going to be hydrophobic. Okay, the head, because it has the negative phosphate group and the positive choline group, is charged. It's polar. Okay, so it's going to be hydrophilic. And that's going to have an attraction to water. Okay, so phospholipids, as I mentioned, are an important structural component of cell membranes. And we have a whole chapter devoted to cell membranes, but let's take a look at how they're going to orient themselves in making up the structural component of a membrane. Okay, so what if you took phospholipids and you threw them in water? So you put them in an aqueous environment. They're going to orient themselves in a very particular way because of their hydrophilic, hydrophobic interaction with water. Okay, so let's identify here. Here I've got a bilayer of phospholipids. So there's a layer here of phospholipids and a layer here of phospholipids. And you can see on this lower layer, they're upside down. Okay, so I'm going to, again, just making the phospholipid models here. I'm just identifying the phosphate and choline ends of these phospholipid molecules because that's the polar end of the phospholipid. Okay, and likewise down here, but they're just upside down. And since water is a polar molecule, they're going to be attracted, these phospholipid heads will have a high affinity for the water, okay, because of their charge. They all have polarity. So these phospholipid heads are all going to have a positive region due to the choline, and they're going to have a negative region due to the phosphate, the negative phosphate group. Okay, and then they're going to have, extending inward away from the water, are their tails. Because they're hydrophobic, they're fearing the water, they're trying to stay away from the water, so they're extending inward away from the water as much as they can. Okay, and we're going to take a look at this in a lot more detail when we get into the cell membrane chapter, but you can see that these tails are extending inward. Okay, so everything in green as far as the tails is fearing the water. So this area in here is phobic, hydrophobic, right? And then the area where the red phospholipid heads are located is going to be philic. Okay, water loving on the two ends because it's attracted to water. Hydrophobic, they're fearing the water, trying to get away from it as much as possible. And so we have, when you throw phospholipids in water, they will orient themselves in this way, and that kind of laid down the basics for how, when cells first evolve, these phospholipid molecules were present in the environment and water was present in the environment, and so there was really the building blocks for making cell membranes already available for life to begin at the cellular level. And so pretty exciting stuff. So those are the phospholipids that we can take from that original group of molecules that we were reviewing. So let's continue on here. Let's talk about the proteins. So on our original list, we had 3 different molecules that I grouped under the proteins. And so let's take a look at those. Each one of these is a monomer group called an amino acid. Okay, so amino acids are the monomers, the subunits that go into making proteins. So we've got one here, one here, and one here. And one structural element that all the amino acids share is on one end they have an amino group, which is an NH2 grouping. Okay, and so we're going to see that here and here, that they all possess an amino group, and then they all possess a carboxyl group. Okay, a COOH grouping. Okay, and oftentimes we're going to see these having charges. Okay, now here's my carboxyl group. I have a carbon double bond oxygen with a hydroxyl group. Now I'm going to just draw this as a negative and just show this hydroxyl group sometimes will donate a free hydrogen to its surroundings. And if this isn't a cell, it's in an aqueous environment. And so anything that donates hydrogens to the solution, you know, is going to take on acidic properties. Okay? The amino group, as NH2, sometimes we'll see it as a plus. Sometimes we're going to see it picking up a free hydrogen proton and forming what's called NH3. And so if it's taking free hydrogen ions out of the solution, we can say it's acting as a base. And the amino group is basic in terms of properties. Okay? And this is something that all amino acids share. They also have what we call an alpha carbon, a central carbon, and then they have a side chain, okay, which we're going to just call the R group. In this particular amino acid, it just has a hydrogen. Okay, that's the simplest of amino acids that exist. Okay, but over here, our R group is this. This is our R group. And on this one, this is our R group. The R groups each make up the remaining part of the amino acid. And there are 20 amino acids that exist in living systems, and they are the building blocks, the monomers that make up all the various proteins in our cells and in our tissues and organ systems and bodies and so forth out there in the living world. They're the most complex, diverse biological molecules you're going to work with. Okay? If we're going to build proteins, we're going to link amino acids together and form our polymers of proteins. Okay? So let's start off simple. Let's build a simple, what we call a dipeptide molecule. And in this example, we're simply just going to link two amino acids together. Okay? So now what we're going to do is we're going to target this hydroxyl group here off this amino acid. So we've got an amino acid here. We have an amino acid here. So these are our monomers. Okay? And then we're going to target the amino group over here, and we're going to target the hydrogen. So we're going to remove a hydrogen, two hydrogens, and an oxygen, and we're going to remove water. Okay, through dehydration synthesis. And then what we're going to end up producing then is a bond between the carbon and the nitrogen. So just ignore this and ignore this because we just lost it as water. And we then form a new kind of linkage between two amino acids, and that's called a peptide bond. Okay, so we've now discussed three different types of linkages or bonds: the glycosidic bonds in sugars, the ester bonds in the lipids, and now the peptide bonds in proteins. Okay, and now we've synthesized something bigger. We've synthesized this molecule, which is referred to as a dipeptide. A dipeptide consisting of 2 amino acids. Okay? Plain and simple. So we're off building a protein. And this is as simple as it can get. So let's go from there and build something bigger. Let's start building a polypeptide. Okay? So we'll do the same thing just like we did with our dipeptide, is I'm going to remove a hydroxyl group and a hydrogen from the amino group and the carboxyl group between the 2 amino acids and form water. Okay? I'm going to do the same here. Take one of those hydrogens, that hydroxyl group, and remove water. I'm going to take this hydroxyl group here and a hydrogen here and remove water. And that hydroxyl group here and a hydrogen here to remove water. And I will now have produced a peptide bond here, a peptide bond here, peptide bond here, and a peptide bond here. Okay? So I've produced 1, 2, 3, 4 peptide bonds. And now I've produced a polypeptide. Poly meaning many amino acids that make up the sequence. We sometimes will equate the term polypeptide referring to a protein. Okay? And we can go a lot further. When we start linking these chains together, we start building our polypeptide, and then we can start talking about the 4 levels of protein structure. But that's for another discussion. That's also discussed in your textbook. Okay? So we can build a polypeptide. There you have it. Proteins. Okay? And the last group here are the nucleic acids. The nucleic acids are going to be our DNA and RNA molecules. Okay? Nucleic acids. These are what sometimes referred to as the informational macromolecules in our cells. There's a relationship between DNA and RNA and the group we just got done talking about. Okay, there is this flow of information that occurs in our cells and every other cell that exists out there. We go from DNA to RNA to protein. Okay? DNA in our cells has all the information on how to make every protein. Okay? So if we use a little analogy, DNA is kind of like a cookbook. A cookbook that has all the recipes to make all the casseroles, all the cookies, all the breads, all the various things that we want to cook in the kitchen. Okay? RNA is simply going to be a message. Okay? This is going to be the recipe. Okay? So it's only going to give us instructions on how to make chocolate chip cookies or how to make a tuna casserole. It's not going to give us all the recipes to make all the different things that we can make. So in a cell, this flow of information is that we're going to make just transcribe a recipe from our DNA cookbook, and that's going to be used then to build a protein. Okay? And so it will be a specific kind of protein. Maybe it's sucrase, which is the enzyme that breaks down sucrose, a disaccharide sugar. Okay? So one specific protein is produced from a single RNA. And the DNA has the recipe to make all the enzymes, all the other proteins that go into our cells. Okay? We identified from our original group, this was a phosphate, this was a sugar, and we specified this was a 1, 2, 3, 4, 5 carbon sugar. Okay, and then what we've got here is this carbon, nitrogen structure here. This is a double ring structure. This is what we refer to as a nitrogenous base. Okay, and there are 5 types of nitrogenous bases, and I'm not going to get into those right at the moment, but anyhow, nucleic acids, both DNA and RNA, their monomer group is something that's called a nucleotide. And a nucleotide consists of a phosphate group, 5 carbon sugar, and one of those 5 nitrogenous bases. So let's take a closer look at what a nucleotide, what goes into making up a nucleotide. Okay, so as I just mentioned, we've got our building block here. We have a phosphate group. Okay, we've seen this already a few times. We have our 5 carbon sugar. And our base. Okay, and I'm sometimes going to just refer to phosphate sugar base, PSB. All right, so all these phosphates are going to link to the sugars, and they're going to come together through dehydration synthesis. The sugar is going to link to the nitrogenous base through dehydration synthesis, and we're going to form our basic monomer group called the nucleotide. Okay, and since there's 5 types of bases, okay, and I'll just simply list them here. There's the A, which is the one that we're looking at here, okay, and then there's T, and there's G, and there's C, and there's U. Okay, adenine, thymine, guanine, cytosine, and uracil. So all the nucleotides, the phosphate, the sugar, well, they won't be the same for everything because there's a difference between DNA and RNA. In DNA, the sugar that would exist here is a 5 carbon sugar referred to as deoxyribose sugar. Okay, in RNA, it's going to have ribose sugar. And in this example, this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. This is the 5 carbon sugar. This is ribose. Off carbon 2, there's a hydroxyl group. Okay, so this is ribose. In DNA, it's going to be identical to ribose, except it's off carbon 2. It's going to be missing that hydroxyl group. It's just going to be the hydrogen. Then that would be deoxyribose. Deoxy means less oxygen. Okay, so we're going to just remove that oxygen from that hydroxyl group in deoxyribose, which goes into making up DNA. Okay, and so both DNA and RNA are polymers of nucleotides. So let's see how that works. So if we want to start building our nucleic acid molecules, we're going to link nucleotides together in chains. And that's what you're seeing here, is we just see an alternation of phosphate, sugar, base. Phosphate, sugar, base. Phosphate, sugar, base. Phosphate, sugar, base. Phosphate, sugar, and base, phosphate, sugar, and base. And so we're now creating something that's called a polynucleotide. Excuse me. And the way that we would link that is we would link the phosphate group of one nucleotide to the sugar of the other nucleotide. Okay? And one of the things your book talks about is that when the phosphate of one nucleotide is bound to the sugar of another nucleotide, if we zoomed in on the sugar, let me do this for a sec. Let me just clear that out. And let's say we zoomed in here. Okay? This phosphate is going to be bound to carbon 3 of the sugar. So we have carbon 1 here, carbon 2, carbon 3. This phosphate is going to bond. This would be an OH grouping. And we're going to remove water through dehydration synthesis by taking a hydroxyl group and a hydrogen. Then we're going to link the phosphate to the third carbon. Okay? So sometimes we're always going to see that. So we're going to see that with every one of these phosphates linking here. Okay? Carbon 3. The bond's going to form there. Carbon 3. This phosphate's going to bond here. Carbon 3. The next phosphate's going to bond there. So when we're looking on this side of the sugar, they're all getting bond to the third carbon. So often we'll say this is the 3' side of the nucleoside chain that we're building. Then if we look at it this way, if we look at the phosphates that actually make up phosphate, sugar, base, right? This phosphate is bound to carbon 5. Carbon 1, 2, 3, carbon 4, carbon 5. So this phosphate's going to bond to, we would bond that phosphate to the sugar through dehydration synthesis, but that's already going to be incorporated in the nucleotide. So we'll say then this is the 5' end, because all these phosphates are bound to carbon 5 of the sugar. So we'll say this is the 5' end, and then down on the other end is the 3' end. And I'm telling that because right now, let's say if we were building DNA, we've only built half of a DNA molecule. Okay, in DNA, the structure of the molecule is like a ladder. And a ladder has 2 sides. And in the middle are the rungs. Okay, so what we've just constructed here is half of the ladder. We've constructed one side. Okay, so think of the alternating phosphate sugar. That makes up what we refer to as the backbone. Okay, let's see if I can just draw a line through here. I'm just drawing a line through the phosphates and the sugars, and this makes up the backbone or the side of the ladder. Okay, extending inward then are the bases, and these make up the rungs of the ladder. Okay, so our bases are the rungs, and so we're seeing alternating phosphate, sugar, and then base, phosphate, sugar, and then base, phosphate, sugar, and base, and so on. Okay, and we'll indicate this is the 3' end. This is the 5' end, because this is representing this side of the molecule. Now, if we had to build the other side, we'd have to, over here, what we end up doing, and this is one thing we're going to find out, is that if, say, this is the orientation 3 to 5' here, on the other side, the other side of the DNA runs the opposite direction. Okay, so that means the 3' end is going to be here, and the 5' is going to be down here. And if that's the case, that means the nucleotides on this side are all going to be upside down compared to what we're seeing here. So I'm going to start here and start with a phosphate, sugar, and then a base. Phosphate, sugar, and then a base. Phosphate, sugar, and then a base. Okay? And so I'll extend these bases inward like this. So I'm finishing off the rungs, the steps of the ladder. Okay, so if I drew the nucleotide, let's say, use green here, kind of like little L's. That's a nucleotide that's linked to this nucleotide. That's linked to this nucleotide. Okay, so on the other side, the way that they'd be oriented is the phosphate sugar base. So now they're running upside down to each other. Okay, and then what links the bases together in the middle here are going to be hydrogen bonds. Okay, and it depends which bases and how many hydrogen bonds they're going to have. Okay, so that's one thing, that DNA has two strands, two sides. It's a ladder. RNA, by comparison, is only half of a ladder. It's only a single strand, so it'll be one-sided with the rungs sticking in like this. Okay. So that's a lot more information, but I could tell you a few more details about, in terms of the difference between DNA and RNA, and that has to do with the base pairing. In DNA, we're always going to see adenine pairing with thymine and cytosine pairing with guanine. And in RNA, we're always going to see cytosine pairing with guanine, but adenine is going to pair with U. Okay, and U stands for uracil. And uracil replaces thymine in RNA. So it replaces the T. And if you look at the structure of thymine and uracil, you'll see they're very closely structured similarly, but they're found in two different nucleic acids. Okay? And we'll specify the relationship between, how we go from DNA to RNA to eventually a protein when we discuss protein synthesis later on in the semester. Those are the nucleic acids, just giving you a brief introduction. And we've spent a great amount of time gathering information about biological molecules and just showing you a few example biological molecules that you could expect to see on an assignment or upcoming exam. You should be able to identify them, know their structure, know their functional aspects, and if you do, you'll be in good shape. You're on your way to understanding a little bit of organic and biochemistry, but most importantly, it's helping you understand biology, the study of life. So thanks for watching this video and this Biocast, and I wish you well.